Welcome to Creative Peptides. Today, we talk about the application of peptides for cancer research. According to research of the World Health Organization, about 12.5% of deaths are caused by cancer worldwide. As is shown from Wiki, cancer is characterized by abnormal cell growth with the potential to invade or spread into other parts of the body. With the development of the cancer, metastasis is the crucial reason that ends up life of the cancer patient. In the past few decades, peptides therapeutics began to bring a promising field for cancer therapy. Recent study shows that peptides play a crucial role in cancer research, including early diagnosis, prognostic predictors, and the treatment of cancer patients. Unlike other therapies, peptides possess unique properties and show their superiority in the therapy against cancer, such as peptide vaccines, which has attracted increased attention from the scientists. The potential use of peptides as anticancer drugs are highlighting current scenario and future prospects. Here, we talk about the following: checkpoint inhibitors, LHRH and analogs, fibronectin fragments, RGD peptides, MMP inhibitors and substrates, somatostatin and analogs, tyrosinase sequences, growth factors and analogs, melanoma peptide sequences. Immune checkpoint inhibitor is a type of drug that blocks certain proteins made by some types of immune system cells, such as T cells, and some cancer cells. These proteins help keep immune responses in check and can keep T cells from killing cancer cells. When these proteins are blocked, the brakes on the immune system are released and T cells are able to kill cancer cells better. Luteinizing hormone-releasing hormone, also known as gonadotropin-releasing hormone or luteinizing hormone-releasing factor, is a hypothalamic neuropeptide which acts on the pituitary to stimulate the release of luteinizing hormone and follicle-stimulating hormone. LH and FSH, collectively called gonadotropins, are involved in the control of gonadal steroids, estrogens in female organisms and androgens in male organisms. The effects of LHRH are mediated by high-affinity G-protein-coupled LHRH receptor on pituitary gonadotropes. LHRH may act as a growth modulatory factor in tumors of the reproductive system. Fibronectin is a large multi-domain glycoprotein that interacts with a variety of macromolecules including components of the cytoskeleton and the extracellular matrix, circulating components of the blood clotting, fibrinolytic, acute phase and complement systems, and with cell surface receptors on a variety of cells including fibroblasts, neurons, phagocytes, and bacteria. Matrix metalloproteinases are directly implicated in almost every biological process involving remodeling of the extracellular matrix, including basement membrane, from embryo implantation to tissue necrosis. In the context of peptidomimetic inhibitors, these compounds bind in an enzymatic catalytic site as the natural substrate normally would. The compounds are derived from the amino acids of the protease's endogenous ligand and are based on the sequences at the substrate cleavage site. Somatostatin was originally isolated from the hypothalamus as the key suppressor GH secretion, but is now known to be widely distributed throughout the body and to be involved in numerous physiological functions. The discovery of 5 distinct somatostatin receptor subtypes has provided an opportunity for creating subtype-specific, and, potentially, action-specific SST analogs. Tyrosinase catalyzes the oxidation of tyrosine to produce melanin and is considered a specific marker for melanoma. In mutant melanoma cells, a misfolded tyrosinase variant is not subject to normal controls or proteolysis. Therefore, increased production of melanin characteristic of melanoma occurs. The growth of eukaryotic cells is modulated by various influences, of which growth factors are amongst the most important for many cell types. A wide range of polypeptide growth factors have been identified and more undoubtedly remain to be characterized. Factors that inhibit cell growth also exist, for example interferons and tumor necrosis factor inhibit proliferation of various cell types. Some growth factors may be classified as cytokines, for example alleles, transforming growth factor beta and colony-stimulating factors. Others, for example insulin-like growth factors are not members of this family. Each growth factor has a mitogenic effect on a characteristic range of cells. While some such factors affect only a few cell types, most stimulate the growth of a wide range of cells. If you need peptides and related services, please don't forget to contact us.